Hey everybody, welcome to yet another journal club brought to you by lifespan.io. I'm Oliver Medvedik, bringing you this journal club from, again, New York City. I don't think I've ever brought it from anywhere else. Trying to think about where I could have potentially brought it from if I was at a conference, but I think it's always been New York City. So welcome everybody. And today we have we have a very basic science, fundamental science sort of journal paper or article. We kind of run the gamut, everything from clinical trials to theoretical papers and review articles, and this is kind of a fundamental research paper. A few journal clubs back, we talked about theories of aging or theories of longevity, and kind of the two kind of extreme theories deal with aging as a programmed cellular event, or a non-programmed event, kind of deriving from entropy. And this paper here kind of puts the ball squarely in the court of a non-programmed mechanism for a main driver of aging. And the paper that we're going to be discussing is Kagan et al. Somatic mutation rates scale with lifespan across mammals. And I believe this paper is from a number of different labs. There's a number of authors, but one of the primary institutes is the Cancer Aging and Somatic Mutation, I believe, Center at Wellcome Sanger Institute in Kingston, UK, along with a number of other different institutes spread across the UK, and I believe is there one in Spain? I think it's Netherlands as well. So, without much further ado, let's take a look at the paper. Share my screen here. Alrighty. So this paper came out recently, and what did they do in this paper? Well, so this is cool in that they looked at a number of different species, and here we have, and they looked at specifically one type of, one grouping of cells that are found in what are in structures called intestinal crypts, which are basically, if you can kind of imagine a glass, so if you imagine something like a goblet, you have a whole bunch of little cells that, and these are all dashed in this area here, that basically are found in kind of on the villi, the microvilli of your intestine. And these cells, the bottom of these cells, I believe you have stem cells, and then they differentiate into cells that line these intestinal crypts. So these are intestinal crypt cells, and they basically perform a lot of functions in your intestines, such as absorbing nutrients, releasing mucus, and so they focused on the mutation, the accumulation of mutations in these cells. So they looked at this one grouping of cells across many species, and they've got the numbers here. I think it was over 50. Take a look in the explanation. 208 individual intestinal crypts. So each crypt, I don't recall how many cells are approximately in one crypt. It's not a whole lot. It's probably, whenever I see pictures of them, it seems like there's like something like less than 100, more than 20. Something like that, from 56 individuals across 16 species with a wide range of lifespans and body sizes. Black and white colobus monkey, if I'm pronouncing that correctly, colobus, cat, cow, dog, ferret, giraffe, harbor porpoise, horse, human, lion, mouse, naked mole rat, rabbit, rat, ring-tailed lemur, and tiger. Right, so a veritable zoo here. So many more species than you typically see in regular research papers, where you stick with organisms that are your model organisms. So this is the histology. This is what it looks like. And they performed genome sequencing and mutational analysis of all of these different species. Now, I just kind of want to, because there's kind of a lot of, genetic and genomic and sequencing data here. I just want to scroll all the way to the end, and there's like a very useful table here, which talks about kind of the variation in adult body size, mass, lifespan, somatic mutation rate, and end of lifespan mutation burden across the 16 mammalian species surveyed. So this kind of captures in a glimpse, like one kind of a summary of what you're sort of dealing with the scales here, and what the correlations are. So if you look at all these animals, the smallest animal was about 20 grams, right? It's probably a mouse, and the maximum 800,000. It's probably a giraffe, right? Full variation is like almost like 40,000, 39,000 difference in mass between all of these different species, and the lifespan ranged from a minimum of around 2.75, maximum of 83.67, which is at the human scale, and full variation being 30. Now, obviously, maximum could be much more for humans. I believe in this paper they kind of rounded it to individuals that 80% of the individuals that made it to a certain age. So just to prevent outliers from kind of clouding the data. So 83.67. So 30-fold variation in lifespans. And the mutation rate per year, this was their key finding in the paper, was 17-fold variation, and the end of lifespan burden was about only a 3-fold variation. So what that means is that when you get to the end of the lifespans of these organisms, you only see about a 3-fold variation in differences in total accumulation of mutations, regardless of these organisms and the full variation of mass or even lifespan. But the mutation rate was 17-fold, and that was the biggest correlation. That was so the kind of the punchline is, before we'll take a look at this data, is that the rate of mutation, the annual rate of mutation, was very strongly inversely correlated with the lifespan of the organisms, right? And this is, of course, looking at somatic mutation rate in the intestinal crypt cells. So, just a summary of some of the data in Figure 1. They were looking at a variety of different mutations, substitutions, so base pair changes, for example, a T to a C, or vice versa. Indels, insertion deletions, which are kind of much larger changes that happen in the genome, so chunks of bases that are missing. And here we take a look at a whole bunch of different organisms, mutations per genome: naked mole rat, rabbit, rat, mouse, now human. And we see that the primary changes that occur are substitutions that occur throughout most of these organisms. And if we take a look at Figure C, what we got here. So linear regression of somatic mutation substitution burden on individual age for dog, human, and naked mole rat samples. You do see a correlation, strong correlation between the amount of mutations occurring with the age of the organism, so over a period of time. So it doesn't matter which, when you take a look at the sizes, which are vastly different, you have this distinct correlation between mutation rate and the age of an organism within that species, right? So the older the human is, the more mutations have accumulated. The dog is, the more mutations have accumulated. So you have a correlation there within the species of mutation accumulation with age. So I'm just going to pause here. We have a couple more figures. Any questions? Okay.
No questions from Facebook so far, but do feel free to jump in.
Yeah.
Folks watching on Facebook or here. And so, yes, you've explained it clearly so well, Oliver, that there are no questions at this moment. Checks in the mail, sir.
Okay, well, I'm sure we could explain it even further. Okay, so mutations. So there's many different types of mutations out there. And just mentioned substitutions, which are changes in individual bases, and much more radical insertion deletions, which weren't the primary mutation observed, but these insertions and deletions were, and they come in many different types. So these are mutation fractions. So this is looking at a human, and these are different types of changes that can occur. C to A, C to G, C to T, T to A, T to C, T to G. So C to T mutations. So there's these mutations cluster in different types of kind of patterns, and there's a whole database of these, and a lot of them correlate with changes that happen in cancers. And there's a database here, and they have basically terms given to them. Let me see if I can scroll up here. Let me stop sharing here, because they mention these SBS1, SBSB, SBSC. And let me just stop sharing here for a moment. Share my screen here. And they mentioned this database here, Catalog of Somatic Mutations in Cancer, Cosmic. And these this nomenclature basically refers to these different types of changes. And the one that I'm just scrolled over here, just randomly happens to be the one that was kind of the main one that was found across species. And this SBS1 is spontaneous deamination of 5-methylcytosine, and this is basically the signature of it if you do a sequence analysis. And there's many others. And they also correlate with, very interestingly, these types of mutations happen endogenously. So the triggers are not so much a mutagen coming from the outside, which would kind of obfuscate things, but more kind of things happening intrinsically within the organism. So spontaneous deamination of 5-methylcytosine causes these C to T substitutions. SBS3, for example, the proposed mechanism is defective homologous recombination, DNA damage repair. I think they also attribute some of the others in the paper, slippage of during replication, for example. Some of them like this are unknown. Then we have ultraviolet light exposure, so on and so forth. Right. So let's go back to the paper here. And I don't know if you're hearing this, but somebody's listening to some techno music in my apartment above me. So if you hear some thump coming through, that's not me. That's my neighbor. Okay, right. So these are the mutational fractions. So we can see at a glance right now that looking at across a lot of different species here, 16, I guess to be exact, you have this C to T substitution, which I believe is SBS1, is the terminology for it predominating. And in different species, there's also a couple of other ones. I think, let's see, what's the terminology for C to A they mentioned there? I'll find that. Is that SBSB? Could be SBSB. There's a couple others. So they look at kind of the preponderance of different types of substitutions, and interestingly, conserved across a lot of these species. The predominant one is this SBS1, which is this change deamination from C to T, which happens primarily in these areas, right, that are called CPG islands, right? So this is where you have a lot of methylation of DNA, and we'll maybe discuss the relevance of that in a few moments. Right? So you have a lot of these changes happening, and these SBS1s, along with a couple of others as well, but this is sort of the main signature. This mutation rate also goes up over time, right? So here they tease apart the particular kind of locus, the particular types of mutations that accumulate. So this is really cool. They really do a deep dive. It's not just looking at kind of gross mutational rates here, or mutations in general, but kind of really splitting the hairs nicely here and really trying to figure out what sorts of types of mutations go up and cluster. And this is really cool because these different types of mutations can be attributed to different types of defects, right? So what do they mention here in the paper? So some of them are called by spontaneous deamination. Mentioned that some of them were due to errors in combination. Well, I'll find that later. It's somewhat buried in the paper. Oh, here we go. So some of the indel stuff is probably a propensity of slippage of template nascent DNA strands. So on and so forth. So there's different mechanistic attributes to these mutations. Okay, so I'm just gonna scroll down here. And stop sharing for a moment. So, any comments?
Yeah, so if we could talk about figure 2 a lot,
yeah,
I agree. That's the really cool part of the paper, and there's a lot going on there.
Mm-hmm. Definitely is.
One of the things which, from my perspective, is really striking is that they have pretty much the same or nearly the same mutational signatures across species, and they get a lot of this SBS1. And one thing which I think is important to think about with regards to this paper is that if you look at this, and they show that SBS1 is a predominant sort of mutation they see across species over age, and you think, well, that's probably the most important type of mutation in aging. And that might be true, but it might be true only for the colon. So the colon is not like all other organs. It's a really, really mitotically active tissue.
Yep.
Which is why, like, if you get acute radiation poisoning, you slough off your intestinal lining, you get acute intestinal damage. If you have mutations in mismatch repair, like if you have Lynch syndrome, you get a whole lot of colon cancer, but you don't get progeria. You get like colon-specific symptoms. So I think one thing that's easy to do is to overread into this figure. Like, it definitely shows conservation of like mismatch mutation across species, which nobody knew before. But that doesn't mean mismatches necessarily apply to like the brain or the blood or the pancreas or whatever. They might, but it's not. It's kind of hard to generalize from one tissue to all in this case.
Yes, I agree with that, and I think DDA had a question. He said only I think we said colon, so maybe he just joined. So yes, and I think that raises a really, really important point, which is basically when I think about aging and differences of aging, there might be some you have this. I recall, a lot of times people talk about, well, there's like sometimes confusion, like, sometimes, in some studies, oxidative damage isn't such a key driver. In some studies it is, but it matters on whether the cells are mitotically active or not. So, I I don't have the references up to the top of my head right now, but I believe oxidative damage is much more important in cells that are not undergoing mitosis. So it could be that, for example, neurons or other non-mitotically active cells have repair mechanisms that really buffer them against oxidative damage. Not so much intestinal crypt cells. There could be other types of mutational signatures that are happening. And I think a key prediction of this would be that, again, it could be that this signature here, is very important, like you said, Michael, for mitotically active tissues. But if we look at non-mitotically active tissues, we might see also a strong conservation across species, but in a different clustering of mutations.
Right.
That basically is and that would be really cool too. And that of course, wouldn't that would still play into the model of non-programmed aging, right? But it's just the mechanisms that the cell types evolved because they're mitotic or non-mitotic. They require different types of longevity maintenance mechanisms. Clearly, telomerase, well, I'm not going to say clearly, it could perform multiple roles, but you would expect for mitotically active tissue, telomerase activity would be important, not so much for non-mitotically active tissue, right? So, so it kind of depends, and so, but it, again, I really like this paper, because this is the first time they've shown this really strong inverse correlation between, looking, the strongest correlation of a mutational accumulation over years here is with lifespan, not like body mass and other things, right? So the strongest, so there was some correlation, I believe, but it was really strong for lifespan, and it was the rate at which the mutations accumulated, not so much the kind of the final amount of mutations that happened at the end, but really, it was the rate that was being regulated or throttled, most likely because of longevity repair mechanisms.
So to make that a little more tantalizing, so there's lots of types of mutations. No one knows what causes many of them, but this particular class of mutation, SVS1, is actually pretty well understood. Like, it's generally regarded this as a spontaneous type of mutation that happens just due to like the interaction of water with DNA. Like, cytosine gets demethylated cytosine gets depurinated. Sometimes you get a bad replacement base. It's something it has nothing to do with like environmental exposures. It probably doesn't have that much to do about with much of anything in the environment. Like, unlike other types, like like tobacco smoke has a lot of mutagens. Radon is a mutagen.
Right,
there's lots of
UV exposure and thymine.
Yes, but this signature isn't that. And the rate, like from a chemistry perspective, the rate of these mutations, you wouldn't expect it to vary so much across species like this paper reports. You'd think, all right, this is something that happens like it's a pure chemical process. You'd think the rate this happens should be conserved. But it's not. It's not remotely. So what are the other species doing?
So I would look at it from a different perspective. Like, let's look at it from a purely chemical perspective. What's the spot? So there might be studies about this kind of the raw kind of chemistry. Now, imagine a situation where you have absolutely no. I'm looking at it from an evolutionary perspective and positing that these longevity mechanisms have evolved to basically buffer the spontaneous deamination from C to D. If you have these genes sitting around, or these sequences sitting around in a test tube with just water at like 37 degrees Celsius with no repair mechanisms whatsoever, let's say you're just kind of running, replication, right, from a PCR reaction or something, what would be the basal mutational rate? And I would predict that it would be like sky high compared to what's happening here, even more so than the mouse, right? So it would be interesting just from kind of a entropic thermodynamic basic like rate of this happening with no other kind of repair mechanisms overlaid to kind of keep this buffer down. And I wonder what that rate is.
Yeah, I don't know. I've never seen papers written on that. I agree. I also think it'd be sky high.
Yeah. So then if something like that was kind of teased apart, then you can then figure out how much of a contribution these different repair mechanisms are laying on top of are kind of doing the buffering. So in that respect, yeah, the changes that occur are kind of really there's like a 16-fold variation, right, among species at the rate at which this happens. But yet, even at the fastest rate here, it might be even way faster, right, compared to the basal rate. So yeah, I'd be interested to maybe there might be some papers going way back when where people basically have done some basic modeling of how fast this occurs, kind of with in just a regular chemical reaction, right?
Mm-hmm.
So it's probably I'm betting there must be some paper from the 1970s or 80s where they just look at they look at spontaneous deamination rates in a test tube. That would be intriguing to compare that to this and see if there can be what kind of information can be gleaned. And I don't know what kind of information we could learn from that. I wonder, I guess, yeah. And they looked at humans, but yeah. Wonder if they didn't look at anything that's longer lived, right? It'd be cool if they looked at a tortoise or one of those sharks that lives 300 years. I guess those are kind of hard to get, kind of rare. So it's kind of hard. It's understandable. Just to see what the theoretical maximum could be, right?
Right.
This could be infinity, but
There is one paper out there looking at SBS1 type mutations in Arctica Atlantica, this really long-lived bivalve with a record of over 500 years. That's the only paper I'm acquainted with.
Mm-hmm.
And I don't know how the rates compare to this paper.
Okay. So yeah.
May I jump in?
Yes, of course.
So, Micah, I'm not sure, why are you surprised about SBS1 being in a much higher rate in, let's say, a mouse? Because it doesn't need to have a low rate. It's going to die pretty soon, right?
I'm sorry. Could you rephrase that? I don't think I understand.
So I'm meaning that the mouse doesn't need to have a low mutation rate because it's not going to survive long enough for it to matter, right?
True. Yes.
So it makes sense that throughout evolutionary time, the repair mechanism would slowly become less and less efficient because it doesn't need to be efficient.
Oh, I completely agree about that on an evolutionary perspective. I think the two... So I guess I made 2 points earlier. One was that there's got to be something that long-lived organisms are doing right to achieve the reduced rate of mutagenesis in colonic crypts. And it's probably just increased mismatch repair. My other point was about tissue specificity, where there's probably different signatures in different tissues.
Sorry.
So with regard to the different tissues, so I put in the chat another paper that says that the mutation rate is similar in neurons. It says neurons and other more mitotically active. It says that the mutation rate is pretty similar. And also looking at it, I see there that there is also SBS1 in neurons. So, okay, but maybe because it's a chemical reaction, so we shouldn't be surprised that we also have it in neurons.
Right. So this is the 2021 Martin Carina paper, which I pulled open from your link.
So if you search for SBS1, so they do say that it does appear in neurons. Yeah.
Mhm.
I'm not that familiar with that paper, but if true, that is evidence against what I said. Although it's also confusing because when I think about mismatch repair pathologies, I think about tissue specificities that don't line up with like I'm not aware of a mismatch repair disease that specifically causes like neuronal deficits. I'm not even aware of the mismatch repair progeria, although I might just not. That might be my fault. Maybe there is one.
Yeah,
it's hard for me to reconcile SBS1 mattering in like neurons, for instance.
Yeah, I'd have to take a look at the paper too. It could be that it's happening, but it's at a location. It's not a key driver of whatever. Oh, anyway, I'd have to.
Thank you for the paper, though.
Yeah.
Sure.
Yeah. So let's take a look at the next group of data here: associations between somatic mutation rates and life history traits. Rates and life history traits. So this is basically where this bulk of the data shows where you essentially have this inverse correlation with lifespan across species and mutation rate. So again, this graph here is these little kind of look like lollipops, but this is mutation rate per year. Each of these are supposed to be different individuals, I believe, or ones that are shaded are different individuals. It's just a way to represent the data to show at a glance. Again, indel mutations are kind of at the low end, and mutation rate you could see, goes up and down with the maximal lifespan of the organism, which they'll show kind of better graphed below. And the end of lifespan burden is, not as much variation like the tables show, kind of correlated. It was a threefold difference, kind of at the end of the, end of the lifespan of an organism, the total burden. So much lower correlation with the kind of the total burden that is accumulated, per genome, right? So I think they controlled for the genome size and the organism's lifespan. So anyway, the fitting is pretty tight. Looking to all of these black dots are different species. So mutation rate per year versus lifespan on a logarithmic scale. It's pretty strong correlation, less of a correlation with the mass of the organism, so how big the organism is. So mean mutation rate per year, log adult mass, kind of a sloppier fit there, and mutation rate per year and lifespan in years, much tighter fit in the model. So a very strong correlation. Between the maximal lifespan of the organism and the mean mutation rate per year. So this is the key finding of the paper here. Is really that. So again, focusing on intestinal crypt cells, which is a plus for this paper because we're looking at a very specific cluster of cells. Where most of the mutations are there's evidence that most of them are endogenous, so versus other types of mutations happening extrinsically through carcinogens and UV light, for example, and looking at it across many different species, right? So this is like really great. And I think, let's see, what's the last paper here? Association between mutation rate subtypes and species lifespan. So here they're basically breaking that same curve down into mutation rates using those, let's see, SBS1 mutation rates per genome per year, SBSB mutation rates. So all of these different mutation rates are basically, have a strong correlation, including the indels, which are more rare, and also, so here they also, they look at mitochondrial mutations, and that is also a strong correlation as well. And let's see, they say, I don't know exactly, it's an extended data, what type of mutations they look at mitochondria, but also appears to show anti-correlation with lifespan. So across species, they obtained an average of 0.23 detectable mutations per copy of the mitochondrial genome by the end of the lifespan, which is mentioned to be a considerable burden because of, it's a mitochondrial genome is really small, 16 kilobases, I believe, in humans, and it's pretty coding dense versus the entire genome. So the implications of this, they go into quite a bit of a discussion, but this is a key prediction of the kind of the random somatic mutation model of aging. That the inverse scaling of somatic mutation rates and lifespan, it's consistent with somatic mutations contributing to aging, right? And with somatic mutation rates being evolutionarily constrained. So there's probably, possibly other interpretations as well, but that's kind of a a main, I guess, kind of the most, I think, obvious interpretation. Now, of course, with these mutations, especially happening in these different kind of clustered areas with changes in, the C, C to T's in the CPG islands, there's a lot of methylation that happens in these areas, and we know that the methylation status of organisms also changes quite dramatically as they age, and it's so, it's the signature is so consistent that you can use that as a clock, right? The methylation clocks of aging, and you can basically, the mutations that happen, the changes are random yet constrained within these areas. So you can use this as, as literally get a biological age for an organism. So, for me, the most obvious next step is to see, how this methylation status changes in response to these SPS1 mutations across organisms, and what the tie-in is there to that. That just seems kind of so obvious. I don't know if they mentioned that in this paper. I can't remember them talking about this, but it just seems like if they did, it was, not. It just kind of flew over my head while I was reading it. And they certainly didn't hammer that point.
Yes, so kind of the fun thing here is that, at least on a test tube basis, this sort of spontaneous mutagenesis that we're calling SBS1, it happens a lot more readily on methylated cytosine than regular cytosine. It's just, I guess I probably can't. I don't think I'm capable of explaining it without having the structures of the molecules in front of me, but there's definitely a dependence of one on the other, at least in a test tube. So you would kind of expect there to be a difference in actual biological specimens.
Yeah.
I think it is required for the mutation. It has to be a deamination of methyl cytosine, right? It has to be methylated first.
It doesn't have to be, because if you depurinate a cytosine, you can still replace it with the wrong base. But yes, the depurination of methylcytosine directly, it becomes thymine right there.
This is the mutation signature. Like when you look at this Cosmic website, they said it's methyl cytosine deamination.
It can also occur if it's not methylcytosine, though. It's just at a much lower rate.
Oh, okay.
Which is why you'd expect there to be a dependence of the mutation rate on whether you've got methylated DNA or not.
Yeah, this is like I'm just thinking about other potentialities and kind of other information that could be kind of squeezed out of this paper. Next steps, obviously, looking at the connection between, so we know that the methylation status changes, right? So is this a key driver of the methylations, right? Is this kind of so one prediction that you can make, one hypothesis is that this spontaneous deamination that happens here, let's obviously all kinds of other mutations are happening as well, but let's just focus on the changes in methylation that underlie the aging clocks and and perhaps drive senescence and so on and so forth. Underlying that, you can hypothesize that this somatic mutation is a driver, and what's throttling that somatic mutation rate is basically the differences in the various repair pathways across species, and probably within species, within maybe humans that have a faster rate of biological aging, from one individual to another. Ignoring the differences from exogenous mutagens, like somebody smoking, for example. Let's just take a look at people that, if you control their control for all other variables, you still have greater biological aging, and that that might be because they have different somatic mutation rates, and the reason they have different somatic mutation rates are because they have differences in their, we'll call them longevity maintenance pathways or repair pathways, and sort of a catch-all term for all sorts of different repair mechanisms. And some of these repair mechanisms are, would obviously, act on certain mutation classes, like what's happening in these SPS1 mutation classes versus other types of classes of, of changes, such as telomere shortening, for example. And there could be overlap between these different types of, pathways as well. Pathways are not always completely orthologous. They could be crosstalk between telomerase and some other pathways. But that's kind of a prediction that you could make based on the kind of the key findings here, right? So then of course, if that's the case, then you can also predict that if you find out what are the differences are in these repair pathways, you could, if your genetic engineering is sophisticated enough and your model is simple enough, then you could tweak those repair pathways to simulate a throttling down and a throttling up in somatic mutation rate, and then seeing that inverse correlation in the lifespan of that organism. So I wonder if you can replicate some of this in simpler organisms like yeast, for example. See, Drosophila is mainly post-mitotic tissues, but I wonder if there's yeast looking at replicative lifespan, and I'm wondering if you can do such experiments to kind of further bolster this. So yeah, just thinking out loud here. And if you could do that, if you could show that by altering those pathways that alter the somatic mutation rate at those locations, throttles the lifespan, not just depresses it. Obviously, you can probably easily shorten the lifespan by screwing up the the mutation repair capabilities, but improve it as well. That would be a really strong indication that that this is where the evolutionary selection is working at that level of at those proteins, at those specific components to regulate lifespan, at least for mitotically active cells, right? So it could be that again, it could be that because even in yeast you get into that complexity where people for many years studied chronological lifespan, which is basically yeast cells that are kind of sitting in their own soup and not dividing in a fridge. And I believe those papers showed that you kind of have this bifurcation in repair, in that cells that are not mitotically active, again, just looking at Saccharomyces cerevisiae, the strain comes from oxidative damage, but not so much when the cells are mitotically active. There's other types of mutations that kind of play a predominant role, which makes sense. But yeah, there's like, I think there's like just a lot of really cool experiments that could kind of flower out of this paper that could bolster, the hypotheses that talk about aging as kind of entropy being a fundamental driver here, right? So I really like this paper a lot. This is my short answer.
Yeah, you could do other things aside from yeast and like Drosophila too. You could do the same sort of experiments you're describing in human cells. Like we can make human colonic organoids, and we can do genetics on them. It would be, in theory, you could like add 5 extra copies of MLH1 and amp up mismatch repair in human cells, and sequencing's cheap enough now where you could do like a month-long experiment and say, all right, did that actually do anything with regards to the mutational signatures? I feel like people have done like positive genetic experiments, like trying to reduce mutational stress, have not had a great track record. Like I think it was the Kenyan lab who like put extra superoxide dismutase into worms and like nothing happened.
Yeah, and but again, it's just so I don't lose my train of thought. What you said there, and what this paper suggests, it really kind of it's like it really brings a lot of things into focus, because it could be it's worth revisiting some of these older papers where things kind of like failed, right? It could be that you're putting in the wrong repair, you're upregulating the wrong repair pathways, right, for the particular type of cell, right? So and then you look at it, and when you look at when you parse out the different repair mechanisms that are responsible for this type of mutation, we might look at that paper and say, well, wait a minute, of course putting more superoxide dismutase isn't going to do anything, because it's not going to affect this type of SPS1 mutation, right? That's driving these cells.
Right. Yeah, I think, what is it? Is it table 1 in this paper that shows like there's only a 3-fold difference in mutational accumulation on death?
Yeah.
Like that's what I think is the cause for optimism if you're thinking about doing like synthetic biology on DNA repair, or like that. It suggests at some point those sorts of experiments have to work. That you should be able to do something to human cells to lower the mutation accumulation rate.
Right. Yeah, it's let's see, what else can I add? I think I've said,
I'll throw in one technical footnote.
Yeah.
So the fact that this paper was possible surprises me. Traditionally, if you're doing like next generation sequencing, the sequencing itself has an error rate, like, and it depends on what sort of how you're doing it. But maybe it's like 1 per 100, 1 per 1,000, 1 per 10,000. If you're looking at like the rate of mutations per cell per year in human, you're in like the parts per billion range. I think this paper worked out to like 100 something mutations per year in their human samples. So the technology error rate traditionally is way higher than the biology error rate. So like, how do you even detect the biology error rate? And I don't know how they did it, but they have a lot in their methods about filtering out technical error. And like, I don't know if this is the Martin Carino lab themselves, or someone else who recently determined how to get rid of the technical error, but you couldn't do these experiments before. You couldn't measure these very relatively trace amounts of biological error in sequencing.
Yeah, we should have
swamp of sequencing.
We should have an expert on for this. Yeah, because that's beyond me. The corrections, that I mean, that's that's something we glossed over here and really didn't discuss. But there is a high degree of technical sophistication to get this data.
I think that's why they use the colon. I mean, there is a part when they say that the colon is like it's very clonal because you have these stem cells, and then they show there is some extended data figure that the variant allele frequency is like usually between 25% to 50%. So they just filter out any variant which is too low, which is a too low fraction, because they assume that the crypt is clonal, and so if you see a mutation, it should appear in many cells in the crypt. And I think that's the reason why they use the crypts.
Yeah, that's one of the main reasons they point out in the paper is because it's understood where all the cells came from, right? So at the bottom of the crypts, you have the crypt stem cells, and so it's basically a clonal population. With when you look at the crypt, it's sort of like a pocket, and then you have at the bottom of it are the undifferentiated cells, and they kind of migrate upwards and get differentiated. But it's it's like a little clonal population, little clonal islands, right, throughout, with stem cells at the bottom. So that reduces a lot of the variation within that kind of cluster.
I didn't think about that. I bet you're right. That must be why they would.
You can find in the comment the right explanation, I'm sure. Anyway, another comment. Oliver, when you said about the SBS1 mutation that maybe drives epigenetic loss, but isn't SBS1 only one-way street? It's like it only removes methylation, right? Actually, I don't know how it works when you sequence. I mean, when you try to check methylation, but instead of C, you have a T. So what's going on? What will they do when we test for methylation?
I don't know. I have to take a look at the data from the other papers to see. It's a good question. I have to take a look at the data and see what the exact changes are, and see if there could be a one-to-one correlation here, or if this is, this is still an intermediary point along the way to changes in the methylation status.
Yeah, the thing with intermediates is that I wonder if it's not a direct effect, then why do we see the correlation with lifespan? Because if there is any node in the middle, any intermediary, then I would assume that this intermediary would be different in different species, and then the correlation would be much worse.
Just thinking here, these areas where these changes have been happening. Let me look through all the supplementary data. So they looked at total mutational loads and different types of mutations, but
they looked at exome, and they showed the correlation stays for exome also when they just consider the exome, and not only for the whole genome. It works also for exome.
Did these changes correlate within similar? Well, obviously they're going to be kind of similar genome areas, but were there specific classes of genes that were affected by these changes?
I think that's something that they didn't. At least they didn't write about it. But also, I think in general, I think the authors, they don't think that somatic mutations are actually a cause, like they actually limit lifespan. That's what it seems.
Well, I mean, how would they? I mean, that's an extremely tight correlation there. So why would they suddenly say that or think that? Let's see. What's this?
So I shared there are like 2 papers with that they also in the supplementary material. I don't know why they put it there, but they in the supplementary material they explain why it doesn't seem like somatic mutations are driving aging, and they say that okay, they do drive cancer, but then there is the problem with cancer and body mass, and it doesn't seem to fit what we see.
It is hard to find a more hotly debated topic in aging than the importance of somatic mutations to driving lifespan. I am familiar with the papers you just linked saying that increasing somatic mutational burden doesn't matter. So again, this is not a resolved topic, so whatever I say is not gospel. A counter argument regarding somatic mutations is that like most, if not, like pretty much all of the human progeroid syndromes where you have accelerated aging are things that directly tie out to increased somatic mutagenesis. You have Cockayne syndrome, you have Hutchinson-Gilford. Like these are things that are either 1 or 2 steps removed from lots more somatic mutations. Some people think that like, okay, you have more somatic mutations, that makes you progeroid, that makes you age faster, but maybe if you have less, it doesn't do anything, which I'm personally skeptical of, but maybe that's true. At the moment, I think I'll have a hard time finding consensus on that topic, which is probably why this particular paper made Nature, since it contributes so much to the what significance somatic mutagenesis has on lifespan.
Yeah,
but I would say that with regard to the papers that I sent, I think that in both of them, SBS1 is like underrepresented. So there are much lower levels of SBS1 in both of them.
Hey, so if you want to be skeptical, remember what I said a minute ago about how this sort of term wasn't possible very long ago. Like there's a certain possibility that you've got technical errors in these experiments because like the naive error rate for sequencing is so high. I'm not expert enough to say which of these papers are more likely to be accurate than others, but since this type of like rare variant detection is relatively new, it's probably good to be cautious regarding any individual paper. I feel terrible saying it because it's like saying, hey, well, half of the papers I disagree with might be bunk because maybe their data filtering method was wrong. But as a non-expert, it seems like there's a lot of software that must go into this sort of data production. And it probably varies lab to lab, and I definitely don't understand how it works.
So I haven't read this paper. I just kind of quickly opened the papers that were just. Thanks for adding that, Oren, to the chat. I mean, so I just clicked on increased somatic mutational burden in normal human cells due to defective DNA polymerases. So these were the somatic mutation rate that was induced by having polymerase epsilon and delta, and this was defective proofreading due to mutations in the exonuclease domain. And I haven't read this, so it says do not exhibit overt features of premature aging, but there's increased risk of cancer. Again, I don't know what types of mutations, again, somatic mutations are, it's a broad category, right? I mean, this sort of like, as we can see from this paper here, you got all kinds of different signatures. It could be that, that this cluster of this type of somatic mutations that are happening from a defective proofreading domain are putting a mutational load on the cells and are a driver for certain cancers, but it might not be the clustering of these types of mutations might not be what you would have as a signature for aging, right? So there could be, you could have an overlap of, so if just sort of, I'm just kind of qualitatively trying to visualize this. If you have a landscape of an entire genome, right, and you have random mutations happening, but they happen randomly within constraints, within different areas and regions, depending on what sort of pathways are broken down. If you have a clustering of a type of mutational load, happening that has a signature that drives cells towards cancer, it might be, these individuals will be dying of cancer and having other problems before you have enough of an accumulation of somatic mutations happening elsewhere that could be a driver for aging. And who knows, I'm thinking out loud here, but maybe some of these cancer-driving mutations, right, like these exonuclease domain mutations, might upregulate, because there's a lot of crosstalk in these pathways, might replicate other repair pathways that say, hey, something's defective here, let me kick into high gear, and kind of perversely, I would say, and again, I'm just making, this is not something that, I didn't do any kind of study here, so, this is just a wild hypothesis I'm throwing out here, but might perversely improve the mechanisms that work on aging, and make somebody age less even though they die of cancer earlier, right? I'm just kind of throwing that out there, just because that would be kind of a wild concept, right? So just because you have a really messed up pathway that induces a lot of mutational load that drives a cell towards cancer somewhere, but then signals other pathways to kind of step up their game, but you basically have kind of not as much repair then happening against the cancer mutagenesis rate, but sort of as a side effect, you have repair happening elsewhere. So that might lead you to conclude that, oh, hey, somatic mutation rate has nothing to do with aging, but it's different types of somatic mutation rate happening in different areas. I'm just throwing that out there. I haven't read this paper. I just read the abstract just now. So here I am wildly hypothesizing based on a couple of sentences I read, on defects in the exonuclease domain of this polymerase, and what that could mean, why that could be a driver for cancer, but not for aging, right? And how I could basically, I'm trying to out loud here reconcile the differences, or the paradox, if you will, right, of somatic mutation rates perhaps going up in some cases but not affecting aging, in other cases going up but affecting aging. So I'm wondering now if you can have, it'd be interesting to see if you could tease those apart. Can you have somatic mutation rates that go up but affect cancer progression and rate, and have no effect or an inverse effect on aging, but other somatic mutation rates going up, affecting aging, but having no effect on cancer rates, right? Can you do these, can they somehow be, can you have this landscape of somatic mutation rates that's simultaneously random, yet sort of siloed a little bit from each other, right? And maybe there's some overlap there that kind of obfuscates and makes things fuzzy.
I mean, if you want an example of siloing, I think one of the more better understood ones are BRCA1, BRCA2 mutations, which are like the mutation that is a clinical marker for cancer risk, and also, so you're a carrier for a BRCA1 or BRCA2 mutation, you're very liable to get breast cancer. Mm-hmm. You're pretty darn liable to get ovarian cancer. That's about it. But it's a double-strand break repair gene, so everything's susceptible to double-strand break repair. Why doesn't BRCA cause all sites cancer? Nobody knows, to the best of my knowledge. For some reason, the effect of that appears to be siloed. Although it gets kind of weird, because if you have two copies of a BRCA mutation, you get Fanconi anemia, like your bone marrow just fails. but just your bone marrow. So again, it's still siloed, just different silos. And I don't think anyone has an answer for why that is, but it is what it is.
Yeah, it's again, I haven't done much reading into it. Could be that the short answer is different other repair pathways kick in and take up the slack, or there's a lot of different things that can potentially account for that. So I think it's worth keeping in mind that but these paradoxes are real, and they kind of need to be reconciled. And I think a model of aging has to account for all of that. Why is it's instead of saying, oh well, here we have a whole bunch of really well done experiments, somatic mutation rate goes up and aging doesn't, and in this case the opposite happens, and I'm on one side or the other. It's like, wait, they might be all right, but we need to reconcile why that's the case. And right now it's sort of my working model is that that, and this paper really hints at that, in that you have different types of somatic mutations, right? And it's just a broad term, somatic mutation. It's just anything could happen. You can have an anvil drop on the DNA, right? Or so there's all kinds of changes that can happen, and depending on what sort of endogenous mechanism it is, a slippage of polymerase, right, that induces mutations in replication, or spontaneous deamination, or it can have profoundly different effects and activate different pathways, right? So let's see. We have a question on Facebook. Did the altered transcriptome deleteriome directly impact aging rate markers? Many mutations do not lead to aging nor cancer. Say, point mutations in non-CPGLs or p53 genes, for instance, or non-regulating gene sequences. I don't know. Did they look at this? Did the altered transcriptome deleteriome directly impact aging rates markers? I don't think they looked at that there, did they?
They didn't look at aging rates, right? They looked at I don't know what aging rates mean, but
yeah,
they just look at somatic mutation, and what I said earlier, the exome is in some extended data, maybe 11.
Let's take a look.
Yeah, it's 11. Extended data, figure 11.
Oh, is there an 11? I think that was a 9. The right paper I have? I think so. Extended data 9. Driver mutation landscape, no issues.
Through the website, you can see maybe here you don't have all of them. Maybe the PDF is not up to date. Through the website itself, you can here you press figure on the right. You see you have sections, figures, references. So press the figures.
Which one? The results you mean? Oh, the figures.
No,
okay.
Figures. Now go down and 11. If it doesn't appear there, okay, it's weird. It's like you're in the other paper. You're not in our paper.
Oh wait, did I open the wrong paper?
Yeah, that was the problem.
Oh, sorry about that. Jeez, looking at a different paper here.
Yeah, but it explains.
Yeah.
It's good.
Oh, here it is. I had the other. It was the other paper that. That was the polymerase paper that you you gave me the link to. Okay.
Yeah, it's my fault. Sorry.
No. Okay, this one. Oh, that's 14. It's a lot of extended data.
11 it was.
Yeah.
But it's just pretty much the same figure like the main one.
This one. So, association between life history variables and alternative measures of somatic mutation rate. Yeah, I think what he was referring to was the maximal lifespan versus the mutational rate. So what was the question here? Yeah, I don't think they looked at the altered transcriptome. Many mutations do not lead to aging or cancer. Yeah. So yeah, I mean that leads to the kind of the statement that it depends, I believe, where these mutations are going to be occurring. That's kind of a fortunate thing with our genomes, is that they're big enough to have a lot of buffering capacity, right? Okay.
I'm still.
Should I go back to the figure? Were you absorbing that figure?
I just am still worried about the fact that the authors think that the somatic mutations don't drive aging, and to me it seems like
Where did they actually say that? I mean,
yeah,
they gave a couple of models that potential, but I thought they were really strongly. I mean, because that's a tight correlation. so it's sort of like kind of not believing your own data. I'm not sure.
Yeah, that's what I'm not understanding. If you go to supplementary note 1, then there the whole note is kind of about that.
Supplementary.
No, you're now in the polymerase.
I got it. Yeah, I know. Sorry. Let's see. So where was this? Supplementary notes? They mentioned this?
Yeah, it's supplementary note
information?
Now. Yeah. Now you need no, it was more up. Right at the beginning. Supplementary information. No, supplementary information. The first one in supplementary information. The first thing there.
This one?
Supplementary information. Yes.
This one. I mean, that's. Oops.
Now it's note 1, but you have to go a bit down.
So here they probably talk about a whole bunch of different theoretical possibilities rather than. I should actually read this, because this is like kind of really interesting, because this is where they start to, go into all the different different theoretical models.
Yeah, but only if you go a bit more, then here you says they are talking about deleterious mutations, and it's like they are saying it couldn't be somatic mutations. It has to be like clonal, clonal proliferation. How do you call it? When clones, selfish mutations become more prevalent. So here, right in the middle here, they are talking about they mentioned the 2 papers that I sent, the polymerase and the other mutation, and they list many reasons why they think it's not the somatic mutation don't limit lifespan. Okay, so a bit up now.
Up here?
a bit up. So now what appears the paragraph in the top. This is the one. So they are like mutations accelerate with age.
Yeah, they mentioned I think their data. So the rate. Yeah. So they say first data across tissue suggest that increase of somatic mutations with again. There's a lot packed in there when it says data across tissues, like which tissues and which areas of tissues, but suggests that or areas of the genomes within those cells increase of somatic with a approximately linear, meaning that somatic mutation rate is roughly constant across life, rather than kind of accelerating at the end, right? I think that's what they showed here as well, but you do have a different rate.
So what's sorry? What's the crux of their argument for why somatic mutations don't appear to affect lifespan? Is it just
okay? So no,
thing?
Don't go away, Oliver. It's here.
No, I'm just scrolling up to the top here to read this.
Yeah, but I mean they're saying like multiple things, and now what you just read, the three first lines are like another issue, and then on the fourth line here they start second mutation rates. Here starts the argument why somatic mutations don't limit lifespan from that second mutation rates in most blah 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 there to the end of the paragraph. I think this is their argument. So they say 15 to 45 mutations per genome is too low, and they show that the synonymous to non-synonymous is not doesn't show that.
Yeah, and that given these considerations, appears increasingly unlikely that somatic mutations could contribute significantly to aging by directly causing cell death or impaired proliferation.
And then they say, okay, if it's not that, maybe it's clonal. Right? That's the next paragraph. Positive selection.
I think what they're going into here, obviously, is okay. So they have this strong correlation. Obviously, it's obvious from their paper they have an incredibly strong correlation between rate of accumulation of mutations and the age, the maximal lifespan of the species. And what these mutations are doing downstream is kind of the problem, where they basically say, all right, well, looking at all these other papers, based on these types of mutations and kind of the amount of them, other data suggest this can't be like the main kind of cause.
I just don't understand how they consolidate what you're saying, the strong correlation. And because I don't think you can say that cancer is the problem, because there are things that, like, if you have, I think whales have like multiple copies of P53, that maybe it helps them, but then we have a different number of copies of P53 than other species, and so the correlation would be broken if you consider intermediaries.
Well, not all mutations are the same. I mean, the thing that they also don't mention here is, again, maybe it's the wrong. Well, they should have mentioned this at least once, but again, how is this changing these types of mutations that they're looking at here, especially considering that the main ones are at the CPG islands? How is this translated to aberrant epigenome status.
They never mentioned CPG islands, right? It's or you're saying it's CPG, it must be CPG island.
Maybe, yeah. I mean, it's so. I mean, is there even a correlation here with a change in, is there a connection with an altered methylation, with the altered epigenome status, with these types of somatic mutations?
The thing is, you also, in figure 4, you see that SBS5 also correlates well with lifespan. Mm-hmm. I don't think that SBS5 is CPG.
Right. I mean, I guess that, again, tying into, well, it's clear. We look at the data, it's clear that there's a very tight correlation here. But it could be that this is, these types of mutations that are happening are just part of the mutational load, right, that happens. It could be downstream effects. I mean, they mention here that the overall mutational burden is insufficient to account for all the damage that occurs in aging. I have to I mean, that's kind of one crude way of looking at it. But if the damage is in critical areas, it could have a kind of a knock-on effect. It could have a much more dramatic role than expected. However, they do mention that the one, and I'm thinking out loud here, the one thing that they didn't notice though in their data is you don't get kind of an exponential increase in aging rates as the organism ages, that it's linear, correct? Which, if repair pathways were being affected, you would expect the rate of aging. Well, is it linear?
Yeah, it's also surprising that the mutation rate doesn't change throughout lifespan.
Because other aspects of aging are not linear, right? Your rate of death and your rate of morbidities is not like linear as you get older.
So maybe repair mechanisms are getting also
your rate of getting Alzheimer's disease goes up dramatically, much more faster between the ages of 60 and 80 than it does between 20 and 40. That's like, I mean, and everything else as well. That happens, right, in a complex organism, right? So kind of at that level, at that macro level, where the damage could be measured physiologically, that's not a linear effect. That's that output as a result of aging is exponential. So, but the somatic mutation rate, albeit very tightly correlated, and being very tightly correlated, inversely correlated to the maximal lifespan of the organism, the rate of mutational rate is correlated, but albeit it is, that in of itself is linear.
Maybe. I mean, what's the strongest evidence that it's linear? I know the paper we looked at today had several species, but for most of those, they were interpolating with only 2 or 3 age groups, and that's not sufficient to affirm or falsify linearity.
Yeah. And of course, only looking at intestinal crypt cells as well, right? So.
I think they also cited some other papers, but maybe those other papers were specifically in human. I don't know.
So if I could respond to another argument that was made, that 4-pronged argument we had up on the screen a minute ago, prong 2, I think, said that there just aren't enough mutations to account for aging, because 15 to 45 mutations a year, how often do you get 2 mutations on the same gene to break it? it would be extremely rare.
Mhm.
And I've heard this argument a lot before, and I think there's a couple of things it fails to account for. One is that in the case of like cancer treatment, where you're giving someone chemotherapeutics or radiotherapeutic drugs, this ages people quite in pretty much every measurable way. It ages them epigenetically. It ages them by pretty much every physical biomarker. And like the classic chemo and radio cancer treatments, they are just mutagens in one way or another. They're things that kill cancer cells, kill all of your cells by mutation. And that definitely causes aging. Now, you might say that's not physiologic because it's way, way too much, but it's still something to consider. So my other counter argument is that 15 to 45 mutations per year. So first off, that's like stem cells. Like I think the lowest mutation rate I've seen in the literature is in the 40s for hematopoietic stem cells. It's a lot higher in cells that aren't stem cells. So like dermal fibroblasts, I think have hundreds of mutations per year, if I recall correctly. But this is also just in the regions we're sequencing. And I think it's really important to consider that some of the potentially most important regions have been unsequenceable until like maybe a year or two ago. And specifically, I'm talking about ribosomal DNA. So ribosomal DNA is kind of upstream of all protein translation. Like your ribosomal DNA is a structural component of your ribosomes. Ribosomes make all your proteins. You have a given human has like between 180 and 430 copies of every ribosomal DNA, which means it's really, really repetitive. There's hundreds of copies every sequence. We can't sequence repetitive DNA. The only way you can do it is with ultra long read technologies like PacBio's HiFi. And like if you look at most like if you do human sequencing, you don't get that information. You don't get the ribosomal DNA information because you can't tell the repeats apart from one another. So ribosomal DNA is kind of interesting because since it's like always being transcribed, the DNA is always open, which means it probably has a higher mutation rate than other DNA, kind of like how heterochromatin has a low mutation rate compared to euchromatin. Ribosomal DNA is like the most euchromatin. It's as euchromatin as it gets. And we can't, when we look at mutation rates, we can't measure there.
It's pretty unstable, and as you know, and a lot of our listeners know, I mean, it's ribosomal DNA. So it's sort of like going back years and years to Lenny Guarenti's lab, right? As kind of one of the first mechanisms posited for a driver of replicative aging in cells and in yeast cells, these extra chromosomal rDNA circles, because they just love to recombine because of their repetitive nature, right? And whether or not that's one of the drivers of aging in humans is yet to be determined, but yeah.
So you can also make a similar but weaker argument about like line and sign DNA. Like this is also unsequenceable. If there's mutations that activate a transposon, that's going to cause a lot of mutational load, and it's basically invisible to us. Although I weigh that less than the ribosomal argument.
Yeah, and also I guess ribosomes you have in all of the mammals, and line and sign I'm not sure. I guess not.
I think they're in. I think line 1 is conserved across all vertebrates, but I'm not sure.
I trust you more than I trust myself on this. Okay.
Okay, I just had a thought here. My math is a little sketchy, so bear with me here. So these figures that they showed here, where they say that the rate of mutations is inversely correlated with the lifespan of the organism, the mutational rate, and then they go on to say that the strange thing is, and I guess that's kind of in their mind strange, is that it's linear, right? And then that can't really correlate for everything, that it's a linear rate. This is like a linear rate in a lot of different, like, and again, let's just say that maybe the data points aren't incomplete here, but if we look at these figures, let's see, oops. we look at all those figures here and all the way to the end here. Yeah. So they're looking at all these rates that are happening per genome per year for SBSB mutations, SBS1, SBSC, indels, mitochondrial. I haven't really, I didn't really, I don't know what, if you took all of these and you kind of, well, if you, let's see what they did here. When they look at mutation rate here, mean mutation rate, right? And I don't know how many sites they looked at for all this, or where all of these SBS1 mutation rates are. So maybe somebody knows a little bit kind of better, or where these indels per genome, or how many regions they looked at. But I'm just going to kind of go out on a limb here to kind of account for a possibility that may or may not be a possibility, in that maybe there is a linear rate of mutations at different locations, but you have more of them happening over time. So the aggregate is not actually a linear increase, but an exponential increase, right? So if you look at different, so it could be a stochastic pattern of linear increases of mutation rates happening across the genome. A few locations that are linear early, more locations that are linear later, and even more locations that are basically just popping up as hotspots. But individually, if you're looking at these areas, they're all a linear rate of mutation. But in the aggregate, if you look at enough of these different regions, you're actually getting an exponential increase over time. Does that make sense?
I think when they say it's linear, they're talking about per genome and not per position.
Right.
I think at the beginning, right?
Yeah, they say the mean, but again, that's yes. So that would kind of contradict what I just said here. But I'm just wondering if in their data analysis that could have been missed somehow.
In Figure 1C, I think it's not mean. It's not. They're not taking the mean. They're showing they're showing it not enough points, like Michael said, but it is mutations, I think, in the whole genome.
Yeah, for what it's worth, I have a strong prior against mutation being linear with age. It seems unlikely to me. Their 1C shows linearity in, I don't know, mouse looks linear. I'd say that like four groupings shows linearity. I find that weird, so maybe I'm wrong. Yeah, their dog and mole rat, you can fit any curve to those, and their human is you can fit a lot of curves to that too. You could draw an exponential through human, since
yeah.
It's not convincing.
Yeah. So it could very well be that it's not that, yes, mutation rate is inversely correlated very tightly, but it might not be, if we kind of if we get more data points in there and look at more regions and kind of really parse this out, it could very well be that it's not really linear. Maybe, I don't know.
If you look at the Cosmic website, I think they say that specifically SBS1 and maybe also SBS5, which is the SBSB. I think they say that they are like clocks, that they are linear. So I think this is kind of accepted, but I don't know. So they also cite this paper. I will send in a link in a moment.
Mm-hmm. Yeah. Again, despite these criticisms and critiques, the authors are listening in. I really appreciate the level of depth that they went into on this, and it's not a critique of the data gathering. It's more of hashing out different potential explanations for some of these paradoxes and questions, right? So, the simplest explanation for a lot of things could be like, well, maybe it's just the data is wrong or something inherent in that, or the cooler explanation is that all of this is real and there's like some really fundamental reason for why you have some of these paradoxes.
So the paper that I sent in the chat, so it's in figure 2, they show, it seems like they start with the 3 mutations that are in our paper, like SBS1, SBS5, and SBS18. And it's like at the top row of figure 2.
In the paper you just, what?
Yeah, I just sent in the chat.
Yeah, this one here, landscape.
Yeah, so the figure 2.
Oh, I gotta subscribe. It's behind a paywall. Oops, that's a commercial for Raid. Yeah,
I can show it.
Okay. Let's see. You want to show it? Or you want to link it there? Great. You got screen sharing capabilities. Perfect. There you go.
Yeah. The problem when you share is that you can't unmute yourself when you share screen. Anyway, so it's the top row here. So SBS1, SBS5 is in our paper, they call it SBSB, and SBS18, it's in our paper, they call it SBSC. And here they have other SBSB, but never mind. So it looks pretty linear, I think.
It's all dependent on those 2 people at the age 40.
It's only 2 people? Oh my god.
Well, it's only 2. I see a purple dot and an orange dot. Like, if you jackknife this, if you if you take the data set and omit single data points, it's not either. Yeah, it depends entirely too much on the intermediates.
Yeah, I don't know. These are nature papers, but maybe I trust the peer review too much.
You can't peer review everything, dude. Like, especially if you do it for nothing. So, especially if like linearity isn't a major claim of the paper, it would, like, if a nature paper claims in the discussion as a footnote that they have linear rates of aging, but they don't have strong evidence for it, it doesn't mean that it's true.
I don't know. It's pretty much. It's like figure 2. It's pretty central.
I mean, that what you highlighted is consistent with the data. However, with what probability? Like, that is a fragile finding because it's that finding is consistent with the data, but it is fragile because it's dependent on a small number of data points. Specifically 2 data points, which are their intermediate ones. Because those are the same people for every mutational signature, right? So it really just boils down to that.
Okay, we need people to replicate. We need people to replicate it, for sure.
I mean, yeah, like, we've been on the line for a while, but if there's one take-home for me from the paper we talked about today, is I want to start doing these sorts of mutation accumulation experiments on my work. I think anyone else working in aging is probably thinking the same thing. Like, if you're in a model system where you actually can measure the rate of somatic mutagenesis, the Martin Carino group kind of showed it's possible, showed that there's some really interesting stuff there. We were talking earlier about like follow-up experiments where we could actually do like experimental tests to try to see if we can manipulate the somatic mutagenesis rate. I think that's where it's at.
Quick question. Is the change in the epigenome status, is that linear?
For sure not. I mean, Horvath's clock is not linear. Right? Horvath's clock, it's logarithmic and then it becomes, right? linear, I think.
Okay.
Okay, but in the end it is linear. It starts logarithmic and then becomes linear.
Hmm.
I thought I had a relevant paper handy, but I don't.
I sent in the chat the figure in the original Horvath clock.
Yep.
Yeah. So at the bottom of this figure,
Yeah, there it is. Let's see. Share this. All right. Yep. Right there.
What's their y-axis? Average methylation.
Averaging average methylation based on training data.
Average methylation on a log scale? Like, are those beta values or those M values?
No, this is, I think this is not the y-axis is, it's not log, I think. I guess it's just the proportion of methylated.
It can't be the proportion because the range would be 0 to 1.
No, you are right. You are right.
This is probably log.
So,
otherwise, you couldn't, like, the scale in it makes sense. So if it's linear on a log axis, then it's exponential on a linear axis, right?
no, I just remember that it ends linear, but it's like weighted average methylation. I don't know why it is -4 to 4, but I really thought I remember it starts log and then becomes linear.
Okay. Change very gradually with age. Of course, the most rapid changes are happening through puberty. Right here. Probably. I'm assuming this is human data. Okay.
So in the text, Steve Horvath says that it starts logarithmic and ends linearly. I copied a quote from the text.
So it is going from 0 to 20. So again, I don't know what sites they're looking at for methylation. I would assume that this is developmental changes, just based on, if we look at, mortality plotted over this. So, these changes might not have anything to do with these highly rapid changes, might not have anything to do with aging. And the ones that are happening here, in this zone, are potentially have a lot to do with aging. Okay. All right. Well, I don't know if there's anything else to add to this paper that we could certainly have a part 2 and go through all the extended data. But I think that was, I think that was a really nice, good discussion we had here. And this is, this is a really nice paper that, kind of deep dives into a very narrow but important topic in kind of aging, in aging research, very basic aging research, very basic fundamental questions. that we need to answer and get the hold on, until then, we're sort of still kind of groping in the dark and kind of cataloging different things. But I think having these correlations with mutagenesis rates and how they, and kind of building a hierarchical tree, what are the fundamental drivers, and does this lead to epigenetic changes, or is there some also feedback going backwards, right? Like, how does this hierarchically stack, and what are all the pathways that I would predict are the ones that evolutionary forces work on, which are the longevity maintenance pathways, and how do they regulate those changes, which are potentially leading more credence to I would say the entropic model of aging, right? These somatic mutations they're looking at here are endogenously occurring, right? And they're happening just because of physics, because of entropy. That's the spontaneous deamination that happens, all these changes, slippage of polymerases. This is not in the classical sense, this is not programmed. This is just what happens at a frequency because of stochastic mechanisms. The only things that can be programmed are the mechanisms that can throttle this down, or I guess in the programmed sense you can throttle it up, but in that sense you're sort of removing the brakes, right, and letting things fall back to their basal rate of whatever the maximal rate of entropy is for the system under those environmental or cellular conditions. Yeah, I think this was a great paper. Answered a lot of questions and also pointed a lot of really cool directions for further research, for us to really get a grasp on all of these threads that hopefully can come together, so we can have a consistent model of aging. And it's heartening to see that a lot of these patterns of damage are conserved throughout a lot of species, right? The rates are different by order 16-fold, but there is constraint within this randomness, right? And I think that's very heartening, which suggests that everything that we're studying in different organisms, the mechanisms for longevity and aging, will overlap with human therapeutics and medicine. And I've got a hand up from Didi.
Yes, to be honest, my problem is always that I'm doing various things at the same time. So I have no time to listen to everything good enough, but it was quite interesting anyway. Just kind of a final question before I'm leaving is, and you can send me the answer if you have an ID by email. My question is there a test on mice that you see that would be possible and useful in the way to understand or even control better the process of aging? Yeah, because at the moment, one of the things I'm doing is try to make a list of possible tests on aging, for some of them to, let's say, support them, to pay for that, and for some of them to place that on a website to try to find people doing that. So maybe, I don't know, maybe with different sort of mice who have different results for this test. I don't know. But if you have an ID, send me an email. If you don't have an ID, don't send me an email.
So I wasn't understanding the question 100%. Is it basically, are you looking for different aging clocks for mice, different ways to measure different rates of aging in mice?
Yes, it could be that. Yeah, well, it could be that, or it could be something else. But I mean, yeah, how to use this data that you have to make some more tests to go further for longevity. Yeah, maybe. Okay, but maybe there is nothing. I try to have that as a question for all times that I'm listening to, because at the end of the day, the goal is, of course, to progress for longevity. So the goal is to see differences and how we can exploit them. Anyway, thank you for all these always interesting discussions.
Thank you. Yeah. Anyway, I enjoyed this paper immensely. What can I say? So there's a lot more, and I have to read the some of the supplemental data because there's a lot of discussion there as well. So kind of getting the authors' insights as to what they think is the key driver here. Because I've got my own ideas as well, but they really yes, Steve wants me to remind everybody to get back on topic, and the topic is Ending Age-Related Diseases Conference. That's coming up soon, not next month, which is July, but the month after, which is August, and I believe it's what are the exact dates, Steve?
Well, you're MC, don't you know?
Yeah, I don't need to know when the exact dates are until the day before the exact date.
See, this is what I've got to work with, guys. He's just the talent, He just rocks up, and that's it. It's us poor schleppers in the background who have got to do all the actual work. Oliver just rocks up,
I just read what's on the teleprompter. San Diego, go.
So anyway, I am of course chastising him in a most jocular fashion.
But yes, we have a very important conference coming up, our Ending Age-Related Diseases Conference, which
August the 11th to the 14th.
To the 14th.
Lifespan Heroes. If you hit me up, I'll sort you out a discount. Oh yeah, because we always do every year. So if you want to get a discount, if you're supporting us as a lifespan hero, such as the fine folks in here are, you can do that. We're also doing a student discount. So again, get in touch with us about that. There's a very generous discount if you're a student and you are interested in learning from the leading experts in the field who are going to be at the conference presenting. So it's a great way to get access, really. So just get in touch with us, and we can sort you out with a code. And I guess keep an eye on the website page as well, as we are adding more and more interesting speakers. And there's more to come as well. We're still in negotiation with some people. I'm going to say that my favourite this year, George Church. I think he is he's always fascinating, and he does so many different things. I'm going to be very interested to see what George is going to be presenting this year. I'm hoping he's going to tell me he squashed the price of genetic sequencing even more, and it's now down to the $50 mark. I know he was on a crusade to get it down to 100, but I think it might drop a little bit more, and he's very keen on that. Of course, George Church was one of the pioneers who was instrumental in the Human Genome Project back in the 80s, and he's done all sorts of interesting things since, like we mentioned last time, the woolly mammoth. Bringing back the de-extincting animals like the woolly mammoth.
I'm expecting him to show us a living, breathing woolly mammoth. Otherwise, I'll be very upset.
I don't know. Maybe we should just save that for next year for the actual physical event in New York, and then we'll have a mammoth burst out through the back of the stage. Although maybe Cooper Union might not like that.
And I better not see a regular elephant with some fake fur glued to it. Better be a legit mammal. I mean, woolly mammoth.
Well, that's scuppered my plan. But anyway, he's a very interesting fella. He's coming along, and we've got a host of other great speakers. So do check it out.
Yes.
And speaking of which, I should tell you where to check it out, which is if you go to lifespan.io conference. You can get there that way, or you can just use /EARD. So either will work. So we look forward to hopefully seeing some of you there. Oh, and also, I even forgot to say, there's even free tickets. So if you'd like to find out what's going on, get a nice high-level view of the event, there are also free tickets as well, which get you access to the keynote for each of the 4 days. 4 days, guys. You can do that for free as well. So
Nice.
What are you waiting for, guys? There's a prize for everybody.
I got a suggestion for George Church. They've sequenced a number of woolly mammoth genomes. I know that they've just recently found a little kind of a infant, or not infant, juvenile one, and they've probably found older ones. You could, in theory, right? If now that we've got these, with the Horvath clocks and the somatic mutational rates that in this paper here, we could figure out what the maximal lifespan is, right? Potentially the rate of aging for woolly mammoths.
It's possible.
I mean, depending on how degraded the DNA was, right? Maybe there's some corrections you can put into there.
Yeah, maybe DNA damage was also relevant to how long they lived as well. Which
how cool would that be to figure out
would be very
the
cool.
Lifespan of an extinct organism?
Exactly. So yeah, I'm very much on the. I am very much on the team. Genomic instability. Is that a team? I'm very much on that team. I do think that it is important in aging. Just to bounce my opinion there on what you've been talking about. If it was not the case, why is it that so many animals that are exceptionally long-lived have extremely stable genomes? I'm just going to say that the naked mole rat is an example of that. The lobster uses, I think it's telomerase, and it's just active all the time, and that also contributes to genomic stability. So there you go. There's my 2 cents. I am very much team genome, and that I've not seen anything that's convinced me otherwise. So there you go. Anyway, that one's going to run and run. I think what Oliver would usually say is more research needed. But good paper. Yeah, good paper.
Yeah. Well, I mean, that's of course I'm going to say more research is needed. That's
and I've got a doozy
biased
for you as well, potentially for next time, which is actually a look at hallmarks. They did a review of the hallmarks and how much they interact with each other. That's an interesting paper, potentially.
Yeah, send me a link.
I will send you a link, and you can decide. But that might make for interesting discussion. How and how much do the hallmarks of aging interact with each other? We know that some, they don't work in isolation, but we know that some impact others more than others. So, it could also be a route to getting more bang for your buck. I'm just going to say team epigenome here on this one, guys. Cellular reprogramming. Yes.
All right. So lots of great speakers. Not just George Church, as cool and awesome as the research George Church does. I don't think he could be the sole speaker for 4 days of a conference.
Oh, I don't know. Have you seen how many projects he's involved?
Yeah, actually he could. If you distributed amongst everybody that worked in his labs, it could easily be a 4-day conference of just a church lab. But
we've got all sorts. Brian Kennedy, Vadim Gladyshev. We've got my favorite, Irina Comboy, who is also now editor-in-chief of Aging Journal. It's going to be a great time, so do check it out. Definitely. And I guess on that note, we'll catch you later, I suppose.
Yeah, and stay tuned for next month's Journal Club, and we'll figure out what the paper is, but it will be, as always, a cutting-edge, cool paper in the field of aging and longevity research. Yeah,
unless we look at something really old, which we have done in the past before as well. But
yeah, unless we do that.
But we probably won't. We'll probably look at something new because it's cool. Right. See you all later.
Cheers.
Thank you.
Till next time.